The overall goal of this procedure is to demonstrate how the principle of PCR thermocycling by thermal convection can be applied to run an actual reaction. This is accomplished by first introducing the idea of how natural convection thermocycling works, along with the basic design principles. The second step of the procedure is to show how to prepare the reactor cartridge and load the reagents inside. The third step of the procedure is to assemble the reactor cartridge into the heater unit and run the reaction. The final step of the procedure is to remove the reagents after the reaction and check the results with gel electrophoresis. Ultimately, results can be obtained that show rapid DNA replication through natural convection PCR thermocycling.
Hi, I'm Radha Muddu from the Ugaz lab at Texas A&M University. Today we will show you a procedure for performing rapid PCR thermocycling using natural convection like the lava lamp. We use this procedure in a laboratory to study the design and optimization of thermocycling devices that harness this principle. So let's get started.
This protocol begins with the design and construction of a simple convective thermocycling device. The device used for this demonstration consists of interchangeable plastic reaction chamber cartridges that are sandwiched between two aluminum plates whose temperatures are independently controlled. Cylindrical reactor wells were embedded by machining arrays of holes in polycarbonate blocks, with different combinations of hole diameter and plastic sheet thickness employed to achieve the desired aspect ratios. Optimal design involves choosing the correct reactor geometry that will